Okay, well then we'll switch over to Carolyn. So the next speaker is Carolyn Pearce, and she joined the Environmental Subsurface Science Group at Pacific Northwest National Lab in 2016, and is director of the Multi-Institutional Interface Dynamics in Radioactive Environments and Materials, iDREAM, which is an Energy Frontier Research Center. Her research focus is chemical speciation, exploiting synchrotron-based techniques to characterize coordination environment and local disorder in phases as they precipitate and dissolve in the environment. She obtained her bachelor's degree and PhD from the University of Leeds in the UK and is a research professor in the Department of Crop and Soil Sciences at Washington State University and a visiting academic in the School of Earth and Environmental Sciences at the University of Manchester. Carolyn?
Thank you, Thomas, for that introduction. Hopefully you can see my slides and hear me clearly.
All good, yes.
Thank you. So I want to start by thanking Thomas, Ashish, Peter, and the workshop committee for the invitation. I guess I did my first synchrotron measurement about 17 years ago at the synchrotron radiation source in Daresbury in the UK. And, that was really my first engagement with the synchrotron geochemistry community, and it's just such a welcoming and friendly and enthusiastic community, and I've loved the years that I've been able to go to the light sources and make these kind of measurements. And it's been incredibly hard for the past year and a half not being able to access the light sources, and so I really appreciate this workshop just to see familiar faces and start to look forward to being able to get back and doing those kind of experiments at the beamline. So I'll start by saying that I feel a little bit out of place in that this is the high temperature geochemistry session, and the work that I'm going to talk to you about today is really focusing on aqueous geochemistry, interfacial chemistry at the interface between minerals and water. But I do feel like a high temperature geochemist today because it's going to be 117 degrees Fahrenheit here in West Switzerland, and so there's no chance for doing anything cool today. With that, I'll launch into the talk. I'm going to take a bit of a different path and talk about soft X-ray experiments. A lot of this work was done by people that aren't me, people at PNNL, and of course the most influential people, which are the beamline scientists at the ALS at beamline 402 and 631, and then collaborators from around the world who've shipped samples and worked with us on these experiments. So I'm going to start with a sort of case study of why it's important to use these techniques to address geochemical problems. And then I'm going to talk a little bit about the technique that I'll mostly be presenting data on today, which is X-ray magnetic circular dichroism, which is a soft X-ray technique which really looks at the electronic structure. So we're exciting the lower energy 2p electrons rather than the core electrons. And so I'll talk through some of the information that you can get from this kind of spectroscopy, and then just launch into a few examples of where we've used them to look at environmental geochemistry problems. Each of them could be a talk in themselves, so I'm afraid I won't be able to give all the details, but hopefully just give you a flavor of what you can do with this technique. And then if there's time, talk about how important it is to link this experimental work with theoretical calculations to understand what you're measuring, and then some more challenging systems where we're moving away from magnetic systems like magnetite, which is much of the discussion up here, and we're getting gradually less and less magnetic. So these X-ray magnetic circular dichroism measurements become harder and harder and more difficult to interpret, ending with some work on ferrihydrite. So to set the scene for the problem, I'm at Pacific Northwest National Laboratory, which is in Eastern Washington State, and it's home to the Hanford Site, which produced 67 metric tons of plutonium as part of the Manhattan Project, and that resulted in a significant amount of contaminated radioactive waste. It's a very large site, and on this site is about 60% of the United States high-level radioactive waste. That waste is in the form of 56 million gallons of liquid, high-level waste liquid, and a mixture of hazardous chemicals that's stored in in these huge underground tanks at the site, which weren't really designed for this long-term storage. And so several of them, particularly the single-shelled tanks, have leaked. And there's ongoing work to retrieve that liquid waste and move it to double shell tanks, which has been completed, and then ultimately transfer that waste to the vitrification plant, where it will be converted from a liquid waste into a solid vitrified waste form for either disposal here on site or at a high-level waste repository yet to be identified. But the contamination that's present has already entered the subsurface, and there's a significant plume of contaminated groundwater that needs remediating, and so there are several remediation activities going on, including pump and treat to extract the groundwater, remove the contaminants, and then inject it back into the groundwater. So this is just showing you what those plumes look like. So this plume is a very large dilute plume of radioactive iodine 129. And technetium, which we're mostly going to talk about today, is shown here in yellow. And you can see that we're, although much of the contamination is in the central plateau at the site, it is migrating towards the Columbia River. And so we need to take mitigating steps to make sure that that doesn't happen. So learning from what's happened in the past and looking to the future, we know that we can reduce risk and uncertainty by really characterizing the signatures of the waste and understanding how it interacts with the environment, and then ultimately developing remediation technologies based on those interactions, and also finding ways to make sure that the waste doesn't enter the environment in the first place. So this risk reduction has to be supported by technically defensible data, and that is really where the synchrotrons come in. This is the advanced light source at Berkeley, which you've heard many talks about already over the last 2 days. So the contaminant of concern that I'm going to focus on today is technetium. It's not present in the natural environment, but it is formed as a product of the fission of uranium and plutonium. And it's of concern in the environment because its longevity. It has a half-life of 213,000 years. And also because it's present as the pertechnetate anion, it's highly soluble. And so it moves readily through the subsurface and into the groundwater and then into the Columbia River, as opposed to other positively charged species that could potentially be like cesium that's attracted to mineral surfaces and interacts with minerals such as clay. Technetium is a major environmental problem at nuclear legacy reprocessing sites like the one at Hanford, where 700 curies of technetium were released into the subsurface. So we need to find ways of mitigating risk to human health by making sure that we can remediate technetium contaminated vadose zones, sediments, and groundwater. To remediate technetium, we need to understand how it behaves in the subsurface. And so we know in the saturated zone, where you have lower redox potentials, you have the opportunity to reduce the pertechnetate anion, where technetium is present in the +7 oxidation state, to this TCO2 solid phase, where technetium is present in the +4 oxidation state. And this reduction can take place as a result of interaction with reduced mineral phases that are present in the saturated zone. There are other mechanisms that can occur in the unsaturated vadose zone, so a much more oxidizing environment, where you have local redox environments at water mineral interfaces, where you can precipitate, and then as the conditions change, you can cover that precipitated phase with an aluminosilicate or a phosphate coating to stop the pertechnetate or the technetium from reoxidizing to form the soluble pertechnetate. So just to summarize, technetium environmental stability really depends on the stability of the immobilized phase, and there is a potential for reoxidation that must be evaluated. But if we can immobilize it and demonstrate that it won't be reoxidized, then we're removing that flux of technetium to the groundwater, and we won't have to remediate it from the groundwater. And so understanding those mineral interactions can perhaps inform long-term technetium remediation strategies in the field, where we can look at things like natural attenuation in the presence of these reduced mineral phases. So to study the reduced mineral phases that are present at the Hanford site, we took some samples of sediments from the Environmental Restoration Disposal Facility, which is a large disposal area in the Central Plateau, and also from the region where the nuclear submarines are buried. These sediments are of basaltic origin, and so they have quite a significant magnetic mineral fraction, about 2–8%. And our results show that this is about 90% magnetite. So what does that magnetite look like in the sediments? These are the natural particles that we isolated from the sediments themselves. And we were able to do some analysis at beamline 20ID at APS to show distribution of iron and titanium, and to work out that these are actually titanium substituted magnetites, so titanium magnetites, where there's about 0.15 atomic percent of titanium within the structure. So using that information, then we went back in the lab and synthesized some synthetic materials for us to be able to determine the redox reactivity of these particles with respect to technetium. We synthesized some micron-sized particles at high temperature, and then a low temperature aqueous precipitation to form these nanoparticles, which would give us some high surface area materials, so we could do experiments on shorter timescales in the lab with that increased surface area to increase reactivity. But we could track back the reactivity to what was happening in the field through these series of microparticles. The technique we use to look at the magnetite and characterize its interaction with the solution and contaminants that are presently in the solution is based on X-ray absorption spectroscopy, which is an elemental specific technique for determining electronic and local geometry of iron or any transition metals in environmentally significant minerals. So it's slightly different from K-edge spectroscopy in that it provides more information on the electronic structure. And certainly for magnetic compounds such as magnetite, it gives you the opportunity to determine not only the oxidation state of the iron, but also the local geometry of the iron and the oxidation state in that local geometry. And so the technique, I'll just spend a few minutes discussing how the technique itself works. It's actually a two-stage process where you first excite an electron from the 2p level of these 3D transition metal ions into the Fermi level, and the excitation depends on the type of synchrotron light that you hit it with. And so at the advanced light source, we have the option of using circularly polarized light, and the sign of the polarization will excite either the spin up or the spin down electrons preferentially. In the second step, the excited electron has to then move back down and find its place in the 3D, in the unoccupied 3D valence band. And that depends on the electronic structure of the material that you're interested in. So it's that that excited electron trying to find its space in the valence band gives you a probe of that valence band structure. This is what the spectra look like. So you take a spectrum with positively and negatively polarized light, and then you take the difference, and the difference is telling you how many spin up electrons you have in the d orbital and how many spin down. And from that, you can look at these complex magnetic minerals like titanomagnetite, where you have iron both in tetrahedral and octahedral coordination and in 2+ and 3+ oxidation. And you can really tease apart those different oxidation states and site symmetries as a function of reactivity. And so this is the reactivity part. These are batch sorption experiments conducted over a period of minutes, which was only possible by synthesizing those nanomagnetic particles to give that high surface area, and then do due diligence to make sure that they were the same structures as we were seeing in our minerals from the site. And this it's magnetite to olivospanel, which is the end member titanium-rich species, gives us the chance to really tune that redox potential of the iron mineral phase by changing the amount of titanium in the structure. So as you add in more titanium 4+ to replace the iron 3+, you have to reduce some of the iron 3+ to iron 2+ to maintain charge balance. And so you're increasing the amount of iron 2+ that's present in the solid mineral phase. And as this experiment shows, that you're increasing the reactivity with respect to technetium reduction by increasing that solid state iron 2-3 ratio. And we can quantify that. So this is that different spectrum that I was talking about in the previous slide for magnetite. And we know from calculations and from the energy position of the peaks that first peak corresponds to iron 2+ in octahedral coordination. This central peak corresponds to iron 3+ in tetrahedral coordination, and then the third peak corresponds to iron 3+ in octahedral coordination. So looking at that magnetite structure before and after reactivity, we can do a mass balance in terms of where the reductive equivalence, the iron 2, is coming from to reduce the technetium. And then we can go back, having conducted that study and demonstrated the importance of the iron 2-3 ratio in the bulk. We can go back and predict whether our material from the site would be able to do that reduction and monitor that over a longer period of time. And again, demonstrate that it's that solid phase iron 2 that's conducting the redox reaction with pertechnetate and reducing it to the insoluble TCE4 oxidation state TCO2. So that understanding of the magnetite structure was just for mineral phases, and showed the application of these soft X-ray techniques for looking at interfacial reactivity. And so we wanted to be able to combine that with laboratory-based techniques that tell us about the bulk iron 2-3 ratio. And it turns out that XRD is very sensitive because of the different relative sizes of the iron 2 and the iron 3 cation to tell you about what's happening with the bulk. And so combining those techniques, we can look at how electrons flow from the interior of the particle to the surface, and then ultimately engage in interfacial reactivity. And these are some examples of looking at that with now biological components that might be present in the environment. So using that combination of XRD for the bulk and then XMCD for the surface, we used this solid solution series of nanoparticles as tunable probes of microbial metal oxidation. And to do that, we isolated this enzyme, this MTOA, which is a decaheme C-type cytochrome, and we exposed that to these iron-bearing minerals that had different solids iron 2-3 ratios, and demonstrated that the C-type cytochrome could directly oxidize metal from the ordered B sublattice of the magnetite minerals at the surface. And we characterized this reaction as a function of pH, and really demonstrated the influence of the cytochrome over just changing the aqueous conditions at that interface with the titanomagnetite nanoparticles. So this was the first time we could really get quantitative insights into these molecular pathways for these biotransformation reactions using just enzymes and these tunable iron minerals. So that was iron oxidation. We also wanted to look at iron reduction, and so we used a cytochrome extracted from an iron reducer, Shewanella-Rhonedensis MR1, and we looked at the OMCA cytochrome. I should say that this work was, this cytochrome work is very challenging, and much of it was done by Jenny Liu, who is a professor in Peking University, but was then at Peon and Helm. And so she did very careful experiments to look at the cytochrome and its redox reactivity. And then we used the XMCD to look at that interfacial region and how the cytochrome was interacting with the mineral phase. And what it showed was that if you compare reactivity just with ascorbic acid as just a chemical reductant, and with the OMCA localizes the reduced ion at the surface of the magnetite, and actually we see from XMCD that there's incipient magnetite formed on the reactive surface because the OMCA cytochrome is able to localize it at the surface and start to catalyze that reaction. This work was also really nicely showing the effective aggregation of particles, and so the smaller particles aggregate more effectively, and the OMCA cytochrome was too large to access the ion in the aggregates. But once you got to a size range of about 30 nanometers, the aggregates had sufficiently large openings that the OMCA cytochrome could get in and access the available ion within the aggregates. So that was looking at oxidation and reduction by separate cytochromes, but this was a study by James Byrne that shows that magnetite can cycle between the oxidized and reduced form, and so it can act as a recyclable electron donor and acceptor for ion metabolizing bacteria in the environment. And so James wanted to study how, when you turn the light on and you stimulate the ion oxidizers during the day, what pool of ion are they accessing in these particles? And then when you turn the light off at night and you activate the ion reducers, how are they extracting or donating the electrons back into the structure? And his work showed by using XMCD and fitting that data that the ion oxidation was relatively easy because ion 2 was localized at the surface, but then the ion reduction was required the conduction of electrons into the particle interior, and so that was much more particle size independent, whereas this oxidation, which was very localized to the surface, was more dependent on the available particle size. And so we can really use this information to understand how microorganisms can access these redox active minerals for growth and respiration over long distances. So I'm gonna, with the remaining time that I have left, I'm gonna switch gears and talk a little bit more about how we combine these experimental measurements with theory to get a greater understanding of some more complex mineral systems where they don't have a very nice magnetic signal, and you only see magnetism through defects or through transformations within the structure. So this is some really nice work by Michel Sassy to be able to interpret the iron 3 L-edge XAS and XMCD, as well as the oxygen K-edge XAS from first principles, using high-level quantum chemical methods, which have some advantages over previous ligand field multiplet models, which were traditionally used to fit these data. And they have some downsides in terms of the empirical parameters that you have to use to best fit the experimental data, especially if you're looking at systems where the ligand field changes. And so these quantum mechanical methods allow us to remove the need for those empirical parameters and really understand what the spectrum is telling us by relating it directly to the structure. So the first example of this is looking at goethite. Computational chemistry suggested that iron 2 3 electron transfer is kinetically inhibited if you have a pristine goethite surface, and so we did some experiments to demonstrate that with Michelle Scherer and Luisa Notini, where they produced goethite using a low temperature method, and we showed from XMCD that this had a slight magnetic signal, which was due to ion defects at the surface, and that this defect goethite could undergo electron transfer reactions with iron 2 at the surface. But when you anneal the goethite and you remove those defects, then you don't get that electron transfer. So goethite, we're getting progressively less and less magnetic, and now we're moving into the realm of ferrihydrite. There's been much debate on the structure of ferrihydrite and its magnetic properties. And this was again a study by Michel Sassy, where he used DFT calculations to predict thermodynamically stable structures for ferrihydrite as a function of composition and water content, and relating that to the magnetic properties. There have been some studies that have looked at ferrihydrite using XMCD. This is one example looking at 6-line ferrihydrite, which is slightly more ordered than 2-line ferrihydrite. And this work really demonstrated from the magnetic component that you could see a tetrahedral iron 3 contribution, and that the magnetism comes from antiferromagnetic coupling between those tetrahedral and octahedral sites. There's also been studies looking at hydrothermal aging over time, and demonstrating again that the magnetic structure of ferrihydrite looks very similar to maghemite, which you would expect, because that is the fully oxidized form of magnetite with no iron 2 remaining in the structure. But we wanted to take it a step further and look at 2-line ferrihydrite over a very long period of time, and what affects the magnetic properties and the structural changes over those long time periods. So we looked at it with both XRD, and showed that the only mineral phase that the first mineral phase that's formed from the transformation is goethite, which you would expect based on previous work. And then over long time periods, you see the formation of hematite, which is predicted to be stable phase at room temperature. So these experiments have been taking place now over a period of about 4 years, and we've been looking at the phase transformation. This is aging under anaerobic conditions. So over the first 100 days, it's dominated by ferrihydrite, and then the conversion to goethite starts to occur, and then it occurs monotonically over these relatively long timescales with the ingrowth of goethite and this relatively constant hematite component. And it just shows that although ferrihydrite isn't the thermodynamically stable phase, because of the kinetics, we are seeing ferrihydrite present in these systems at relatively long timescales. And we can look at the magnetic signature of the ferrihydrite, which is a challenge to measure, because we have to measure these at 20 Kelvin, and the signal is very, very weak. So we have to take many, many measurements to actually get these very noisy data. And we showed that the tetrahedral component is present in the very early stages, but it rapidly disappears as you see the structure of the ferrihydrite changing and it converting to goethite. It's also correlated with the concentration of hydroxyl groups in the structure that are actually acting to stabilize that disordered tetrahedral site. And a number of things will influence it, including whether you age under aerobic or anaerobic conditions, whether you add in iron 2 during the aging process, or whether you dope with cobalt or nickel as a non-redox active proxy for iron 2, to understand where they might be affecting the structural transformations. So I just like to close by saying I hope I've demonstrated the need for using multimodal synchrotron and lab-based experimental approaches in combination with theory to really understand these nanomaterials and their interaction with the environment. I think many of the talks today have emphasized that theme. I'd like to acknowledge the sponsors for this work, and particularly the Advanced Light Source that have supported many, many years of XMCD measurements. And finally, thank you to you for your attention.
Right, thank you very much. There are a couple of questions in the chat that I think we can go to. The first one's from James Green. Does the microbe outer membrane cytochrome transfer electrons extracellularly via nanowires that transfer electrons from the magnetite to the TC, and then encapsulate the TC via the nanowire EPS polymers?
I think there are multiple different mechanisms, but for us, it was really, sorry, I'll just stop sharing. The mechanism was really that direct interaction with the C-type cytochromes and the mineral surface, and we showed the need for that direct interaction with this this careful study of aggregation, where if the cytochromes couldn't directly access the iron 2 because the size of the pore spaces in the aggregate was too small, then we didn't see any electron transfer. But that's not the only mechanism, right? That was just the one in our study.
Okay, and then Paul Fenter is asking, have you tried to use electrochemistry to derive the insertion of titanium into magnetite? Can that be done?
So we haven't tried electrochemistry to derive the insertion, but we can insert titanium into the structure. It's just there's a limit to how much you can get in under ambient conditions. So for the nanoparticles, we could only ever get up to a substitution of about X equals 0.35. And to get to ovospinel, you have to go to temperatures approaching 1,000 degrees Celsius. And the limiting step is really that once you get to the point of having to have iron 2 in the tetrahedral site, that's where you really require the higher temperatures. But that's an interesting thing to think about, Paul. Thank you.
Okay, thank you very much, Carolyn.